So when discussing meiosis, it actually goes through two different stages. So meiosis 1 occurs after interphase, which is the same cell cycle of interphase that happens in mitosis. So this is what the cell is going to look like with the cellular growth, as in G1, the DNA that's duplicated that happens in S phase, and then just the preparedness for eventually separation that happens in G2. This is what the cell is going to look like. It still has the nuclear envelope, the centromeres, then the DNA that's been duplicated. This is the start of meiosis 1, so it's going to start out in prophase 1, which is going to have the homologous chromosomes pair up, and it's going to form these tetrads. So this would be a tetrad, and this would be a tetrad. So then the chromosomes are basically going to begin to condense, and then there's a synapsis, which is the pairing of the homologs, which is what this is right here. Another thing that happens in prophase 1 is the spindle fibers are going to start to form, and then crossing over can actually take place. So what crossing over is basically the trading of pieces of chromosomes between a non-sister chromatid. So that means like these two, since they are not a part of the same chromosomes and they're non-sister chromatids, they can basically trade pieces of the DNA. So the other thing that happens in prophase 1 is when the nuclear envelope is actually going to break down. So this is it breaking down. So there actually next would be a pro-metaphase, but it's not really talked about that much. So we're going to move into metaphase 1. So in metaphase 1, this is when the homologous pairs of chromosomes are going to line up on the metaphase plate. So next would be anaphase 1, which is when the homologous chromosomes are going to separate and move towards the opposite poles. So they're doing this with the spindle fibers that are drawing them back from where the metaphase plate was and drawing two chromatids of each homologous chromosome over to each pole. Next in meiosis 1 would be telophase 1, which is when the chromosomes arrive at the spindle poles and the cytoplasm starts to divide. So this would be representing the division of the cytoplasm, and then the nuclear envelope that's not actually fully there in telophase yet, but it is actually starting to form around each gonna be new cell. So after telophase 1, the next step is going to be cytokinesis in the meiosis 1 stage, and basically what's going to happen is the chromosomes are not going to be condensed anymore, and the cytoplasm is going to divide into two daughter cells. And then, as you notice, the parent cell had 4 chromosomes, and now each daughter cell is only going to have 2 chromosomes. So the next stage is going to be prophase 2, starting meiosis 2, basically. So in prophase 2, the chromosomes are going to recondense, and the spindle fibers are going to start to develop. This is also where the nuclear envelope is going to break down in prophase 1. Next is going to be metaphase 2, which is when the individual chromosomes are going to line up on the equatorial plate. So it's right down the center like this, and these spindle fibers are going to attach to them. So the next part of meiosis 2 is going to be anaphase 2, and this is when the sister chromatids are going to separate and move towards opposite poles. So as you can see, these two sister chromatids separated, and they move towards this pole and this pole, along with the rest of both cells. So next would be telophase 2, which is when the chromosomes are going to arrive at the spindle poles, which we've already seen, and then they're going to start to have the cytoplasm divide, which is what this is representing, along with like the nuclear envelope starting to reappear. So lastly, these cells are going to go through cytokinesis, which is going to be them dividing completely into their own cell. So there's going to be the end product of 4 haploid daughter cells, and they are not going to be identical.